Welcome to Biology Made Easy. Biology Made Easy are biology lessons designed for high school and senior secondary school biology students. In our first lesson, we discussed the term biology. We also looked at the characteristics of living things. Every living thing must have three basic characteristics. DNA must be present, 7 life processes. All the 7 life processes do occur and should have a cell. We started discussing what a cell is, and we didn't finish, and we are continuing cell structure and functions today. You had a homework, and we want to discuss the homework. You were supposed to define life process. How did you define it? Let's see. Life processes are actions or activities that occur in every living thing which keep the living thing alive and ensure the continuation of the species by producing new individuals or offspring. Activities of action or actions. You also had to write down the importance of sensitivity. What did you get? Sensitivity. Right. One, sensitivity makes the organism aware of danger and so escapes danger. It also allows the organism to be aware it needs food, water, nutrients, and so sensitivity allows the organism to go for food or nutrients. Then also it makes the organism look for mate, goes for a mate to be able to produce offsprings. And then it allows the organism to sense or know the importance of looking for a suitable environment. If the place is hot, sensitivity allows the organism to know it and to move from the hot place. So that is it. You notice that the importance of sensitivity, it also ties in with importance of movement, isn't it? One moves to escape danger, to look for food, for mate, and suitable environment, especially when locomotion comes in. All right? Neat. Now let's get on to today's topic. Objectives for the day is to continue. By the end of the lesson, you should be able to describe the structure and function of generalized cells. You should also be able to work out the differences between plant cells and animal cells. 2 main objectives. In our last lesson, we started looking at the structure of cells. We discussed the cell membrane and then cytoplasm. We want to pick up from there and look at the other structures that make up a cell. We looked at the cell, simple structure, cell membrane, with cytoplasm inside, with structures suspended in it, and we said the structures suspended in it that are membrane bound are organelles. Now we are going to look at the structures suspended in it or dissolved in it. All right? So we are going to look at the structures suspended in it. want to look at the mitochondrion. Mitochondrion. The plural is mitochondria. It's a very simple structure. It's double membrane bound. The inner membrane causes it to be thrown into folds. That is it. And it's all talking about how structures are folded. So that's the mitochondria. It's tiny, spherical or sausage-shaped, double membrane bound, inner membrane thrown into folds to increase the surface area, all right, for reactions. Now, the function of the mitochondrion is a site for respiration, all right, respiration inside the cell, all right, and the inside has enzymes. On these folds too are enzymes. All of it helps in the process of cellular respiration, respiration that goes on in the cell. So that is the mitochondrion. Now let's quickly look at another structure. And this time we look at ribosome. All right? The ribosomes are very tiny structures. They are not membrane bound. The whole structure is made up of two things: RNA and protein. No membrane bound. The RNA is ribonucleic acid. See it? We'll talk about the RNA and DNA, all right? It's the ribosome. It's either floating freely in the cytoplasm or is bound to another structure called endoplasmic reticulum. Now, the structure ER, simply endoplasmic reticulum here. All right? The endoplasmic reticulum is simply membrane-bound channels. So if you have a channel, channels, channels, channels. So this is a channel or canal that is bound by a membrane. That's the ER here, membrane-bound canals or channels. Now, these membrane-bound canals, some of them are connected to the nucleus. So if there's the ER, and I put ER here, there are channels. Some of it is connected to the membrane around the nucleus in animal cells. And on these membrane-bound channels, ribosomes are attached. So if this is a red ribosome, it's attached. Good. There are two types of ERs, endoplasmic reticulum. Here, two types. You have smooth endoplasmic reticulum and rough endoplasmic reticulum. The smooth endoplasmic reticulum, the channels, do not have any ribosomes, but the rough endoplasmic reticulum have ribosomes stuck on their outer surfaces. And part of it, if it's an animal cell, the channel can be connected to the nuclear membrane. Sometimes and often, the channels are also connected to the cell membrane and also to other organelles. So if we want to make it simple, and this is the channels are ER, some of the channels connected to the nuclear membrane, some of these channels will also go channels and connect with the cell membrane. Some of the channels are connected to other organelles. That is the endoplasmic reticulum. So if channels, a structure has all these membranes and channels, what do you think its functions will be? Think, channels, channels, channels. One of its function is that protein and other substances are transported in the channels to other areas of the cell. You see that the ribosomes, they are sites for making proteins, ribosomes. So the channels are conducting channels for substances in the cell. All right. Then also another function of ER, we see that it separates the cytoplasm into compartments. So if these are ER, this is one compartment. This is another compartment. And many ER are separating the cytoplasm into compartments. Very neat. So that processes can take place in those compartments. All right. Then another function of the ER is that it makes the cytoplasm solid. Plenty of it, can you imagine, will make the cytoplasm firm and solid. Meanwhile, the rough ER, protein synthesis is going on in there. Then there's also another structure in the cell called Golgi body. Here, Golgi body, they're just stacks of membrane bound flattened sacs. So membrane bound flattened sacs. Membrane bound flattened sacs. Something like this. Flattened sacs. And it has tiny, tiny membrane bound structures called vesicles at their sides. So this is a Golgi body. Golgi body. And it's also inside the cell. Now, the vesicle, these tiny, tiny structures, is a vesicle. These are vesicles. So we have Golgi body, we have vesicles. And these vesicles are single membrane bound. The Golgi bodies also transport substances. Not only that, substances that are simple are built up into huge substances, macromolecules. If the molecules were tiny, micromolecules, they come here and they are built into macromolecules in the Golgi bodies and transported. You see these vesicles? These vesicles contain substances and they are like transporting sacs. They will move from here to another part of the cell. So here we have Golgi body. Golgi body is here with the vesicles. And the vesicles, they cut off and are moved to other parts of the cells. Very neat. Transporting sacs. That's Golgi bodies. Good. Now we want to look at another structure. And these ones, we want to put them together. Single membrane bound fluid filled sacs. There are three types we want to look at: vesicles, lysosomes, and vacuoles. Vesicles are tiny, very tiny, you see, formed from the Golgi bodies. All right? And they are transporting sacs. They transport material. Lysosomes are also just a single membrane bound fluid. All right? That's a lysosome. The lysosomes too are built from the vesicles. But the lysosomes do not only, they contain enzymes. All right? So lysosomes are bigger vesicles. They contain two types of enzymes. One type of enzyme is able to digest old organelles. So if this is a lysosome, it does two things, two major things. One of it contains enzymes to digest old organelles, bacteria, and foreign matter. And that process is called autophagy, all right? Then this lysosome also has enzymes to digest the whole cell. The whole cell will be digested. And that is autolysis. So now let's look at vacuoles. We've looked at vesicles, lysosomes. Now I want to look at vacuoles. Vacuoles are big. Big, very, very big. They are tiny in animal cells, but in plant cells, they can be big. This whole thing is a vacuole in a plant cell, big. And sometimes in plant cells, the vacuole can fill almost the whole cell. We'll come back to this. Now, what is the importance of vacuoles? In animal cells, vacuoles serve as transporting sacs. They are tiny. Food vacuoles in amoeba, etc., prototists. Vacuoles are food vacuoles. The vacuole contains food. They can also be vacuoles that do osmoregulation. Osmoregulation means if this is a vacuole, it takes in water from the cell, it moves to the surface of the cell, bursts to release the water. That's in lower animals, like lower prototists, amoeba, etc., etc. Osmoregulation. That's in animals. All right? Now, in plants, the vacuoles store substances. The vacuoles will store a lot of food sugars, water, etc. All right? In plants, vacuoles are very, very important. Osmoregulation. All right? The vacuoles store water. The vacuoles make the cells turgid, firm in plants. All right? And in plants, the vacuoles store pigments. So sometimes when you see a plant flower, red, etc., the pigments, the color is in the vacuoles. All right? So that is, and the vacuoles make the plant cells, especially when this plant is herbaceous, not thick. It makes the cells firm and turgid to keep the plants stand firm. Those are vacuoles. Now, the substance that fills the vacuole in plants is called cell sap. So you have a vacuole here. Its contents is called cell sap. You have it here. Good. Now let's quickly look at chloroplast. Chloroplast is also found only in plant cells. And chloroplast is also another structure, double membrane bound. The inner membrane is thrown into stacks, form stacks, 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 stacks. All right? And the chlorophyll, which is green pigment, is found in those stacks. So simply, the detail will be done some other time. That's a chloroplast and is a site for photosynthesis. Then we have cell walls in plants. You have this cell membrane. This is a cell membrane. And in plant cells, you have the cell wall. All right? Okay. Now, plant cells are drawn, not the end. You have the cell wall. Now, this cell wall is made up of cellulose. All right? It's made up of cellulose in plants. It's made up of chitin in fungi. It's here. It's the outermost layer. It has pores in it. It's freely permeable. It allows everything to go in. All right? The function of the cell wall is it makes the cell firm, assists the cell to be firm. You know, plants sit at one place, so if they are not firm, they will be contorted. So the plant cell makes the cell, gives rigidity to the cell. It also takes part in osmoregulation. Osmoregulation, regulating the amount of water it will not allow the cell to take in so much water so it doesn't burst. That's the plant cell. Very neat. All right. It doesn't allow, so excess water cannot move in. Now we want to talk about middle lamella. It's also a structure in plant cells. You have a plant cell. Let's say we have these two plant cells. These are other cells around because plants, so we have these two cells. We have here, this is the cell membrane. This is a cell membrane. This is a cell membrane for this cell, all right? And then the cell wall for this cell is here. For this cell. Now this cell, here is this cell membrane. And its cell wall is also here. Now, middle lamella is cement, calcium peptide. Is a cement that is found in between the two cell walls. So this is middle lamella. That's a common cement layer cementing the two cells. Very neat. Everything that goes to make plant cells rigid is done. Now, you see the chloroplast has chlorophyll. There are other structures called plastids that store things like starch is formed in a plastid. All right, so there are other grains, you can call it, in plant cells. And then there's this structure, cytoskeleton. Cytoskeleton, this structure. It's kind of protein material, all right, like protein filaments, all right, that can contract and relax like a muscle. So this cytoskeleton are tiny fibrils, all right, in cells that can contract and relax, all right, shorten. There are so many other things in cells. So quickly rounding off, this is a generalized animal cell. We have, oh, we didn't talk about the nucleus. The nucleus is double membrane bound. All right, it doesn't have only one membrane around it. Quickly, the nucleus. How could we have forgotten the nucleus? It has its membrane, double membrane bound. So the membranes around the nucleus is two, double membrane bound. All right, it has very tiny pores. So this is the nuclear membrane. Nuclear membrane. And it contains fluid called nucleoplasm. And you know it has the DNA inside. All right, the DNA is also inside it, but the DNA is normally seen as chromatin material. But when the cell is dividing, then you see the DNA real properly. All right, that seems to have a space around it, kind of material that forms a clear space and is called nucleolus. This is the nuclear membrane, is double membrane bound with pores. Here is the nucleolus. Here is DNA or chromatin material. You know the function of the nucleus, isn't it? All right. controls all the activities of the cell because of the presence of the DNA. What about the function of nucleolus? It forms RNA, ribonucleic acid. The ribonucleic acid, you see the DNA, it can't move out of the cell. So it gives its information to form RNA. And the RNA moves out of the nucleus and it causes proteins to be formed, depending upon the information, genetic information it picked from the DNA. Very neat. We'll talk in detail about this in a good time. So that's the nucleus. It has its own cytoplasm, nucleoplasm, and things are in it, dissolved. Serious matters are going on. Okay, now quickly, as we round off, let's quickly look at the difference between animal cells and plant cells. You should be able to list them, isn't it? Quickly, what do you say? Cell wall? No cell wall? Neat? Chloroplast here? No chloroplast there? All right. This animal cell has something called centrioles that plant cells don't have. Let's look at centrioles and you go and find out what centrioles are. Centrioles. All right, centrioles. Find out what centrioles are. All right, they are very close. Found out close to the nucleus. Some rod-like structures found close to the nucleus. Concerned with cell division. That's centrioles. Okay, right. Animal cells have them. Plant cells don't. Plant cells, large, permanent vacuole. Animal cells, tiny, temporary vacuoles. What else? Have we left anything out? Have I left anything out? If I left anything out, please remind me in our next lesson. All right? Plant cells, animal cells. As I said, as the plant cell, when it starts as a cell in the embryo, it's a very simple cell. But as the plant cell grows, the vacuole grows. And in some plant cells, the vacuole fills almost all the plant cell, pushing the cytoplasm and its contents to the sides. Well, we've had a good day with cells, isn't it? And it's a continuation of looking at characteristics of living things. So for your homework, what are the relationships? Is there any relationship? Is there any relationship between cell structure, the cell, and the 7 life processes? Good. So till we meet in our next lesson to discuss biological techniques, we need to know the techniques that a biologist needs early before we go on to talk about other things. So in our next lesson, we'll look at biological techniques, what a specimen is, sectioning, symmetry, and orientation. So till we meet in our next lesson, have a good day. Bye-bye.